Hello and welcome back. Today I'm going to be talking about red blood cells. We're going to be talking about their function. We're going to be talking about their structure. We're going to be talking about blood typing and blood transfusions, as well as we're going to talk about some research on potential synthetic hemoglobin and oxygen carriers. My name is Matthew J. Greer. This is a presentation prepared for Biology 520, Concepts of Anatomy 2, through Grand Canyon University. My professor is Dr. Sandra Barrera, and today is the 19th of January the year 2022. Without further ado, let's get going on this thing. So the erythrocyte, or the red blood cell, is a disc-shaped cell inside of the body, and here you can see this little concave disc here. And like everything else in the body, the structure of this cell is going to determine its function. So it's disc-shaped so that it has the maximum amount of surface area. And surface area is very important for these guys because they carry vital oxygen to the cells and they take carbon dioxide away from the cells. We need to be able to get this oxygen into our cells for cellular respiration, for the Krebs cycle, more specifically in the Krebs cycle for the electron transport chain part of that, where we're actually taking the glucose and we're taking all those high energy molecules and we're using those hydrogens to move that electron pump. They lack organelles and a nucleus. This is also to maximize how much they can carry, how much oxygen they can carry, and how much carbon dioxide they can carry away. They have a flexible membrane. When we talk about the capillary inside of the body, these blood cells need to go through those capillaries and they need to be able to bend and fold and go through them in really, really small spaces. Not only so small even that there's one cell at a time going through and there's not enough room for two of them to go side by side. The outcome, the perks of all of this structural things is we're able to fit 250 million hemoglobin molecules per RBC, per red blood cell. And this is really awesome because everything in our body needs oxygen. The brain needs oxygen, muscles need oxygen, the liver, the pancreas, everything needs oxygen. So it's good that we have these nice, highly developed transport systems that have gotten rid of all of their organelles, nucleus, to maximize their surface area and be able to carry the most amount of oxygen. So to carry oxygen, red blood cells are going to use hemoglobin, which is this molecule that is designed with two parts. It has a globular protein part, which has both alpha and beta subunits that are a little bit different from each other. It has a heme group, which is located in the middle of these subunits, and the heme group is where the oxygen actually binds on. So each one of these hemoglobins has 4 binding sites, can bind 4 oxygen. So those 250 million hemoglobin per cell, we end up with 100 million oxygen molecules. Hemoglobin also, because of this structure that it has and this iron that it has and this ability that it has to carry gases, can carry things like carbon dioxide. It can carry the hydrogen ion of carbonic acid, and it's really important in the buffering system, keeping that pH stable. It can carry carbon monoxide. And carbon monoxide has a higher affinity than things like oxygen. So if there's oxygen and carbon monoxide present, that hemoglobin will carry that carbon monoxide. This is important when we look at cases of carbon monoxide poisoning. This is why it occurs. And nitrous oxide, which is a vasodilator. It helps expand the blood vessels, lets things move around in there. All of these different things can be carried on hemoglobin, in addition to oxygen, regular O2. So red blood cells, the bone marrow is where they start their life cycle. In adults, our red bone marrow is primarily inside of our axial skeleton, so head, ribs, clavicle, hip bone, and then it also is inside this epiphysis of these long bones, both up here in the humerus area and in the femur. In children, they have much more red bone marrow. As you get older, that goes and transforms over into yellow bone marrow in the long bones, and is not as good anymore and incapable of producing these red blood cells. Now, these red blood cells are created through the use of pluripotent stem cells. So these stem cells go and they make new red blood cells, but they also, through some additional processes, can turn into white blood cells. They can turn into lymphoblasts, which then go to white blood cells. They turn into the platelet part of blood, and they make the red blood cells, the part that carries oxygen. So these pluripotent, meaning that they have more than one outcome, stem cells, are really important for cardiovascular health and for the circulatory system. Once these guys have matured and they're now ready to do their job, these are allowed to enter the bloodstream. And there are some instances where immature blood cells can be introduced into the circulatory system, and we'll talk about those. So what determines when we're going to make these red blood cells? Well, low oxygen, the thing that is carried on side of these red blood cells, probably means we don't have enough of them. So this triggers this process of erythropoiesis. And erythropoiesis means making blood. So the kidneys, not the bone marrow, the kidneys is what actually senses these oxygen levels. And they are going to release erythropoietin, which is abbreviated EPO. So real quick here, let me sketch this down. Low O2 is going to stimulate these guys, stimulate this kidney, produce this erythropoietin, which travels to the red bone marrow, and it says, hey, we need to release more red blood cells because we need more oxygen. Oxygen is important for the body. After that process takes place, which depending on how much oxygen we need, it might be more red blood cells made. So these red blood cells are now made and released. They will now carry oxygen. So oxygen normal. Oxygen good. We have enough of it now. This will cancel this production of erythropoietin in this kind of negative feedback loop there. If there is a reason, let's say a hemorrhaging or a large loss of blood, reticulocytes or immature blood cells can be used and will be released as an emergency reserve. These still have some ribosome fragments in them and remnants, but in a pinch, they will work for carrying oxygen. So your body will release those in an attempt to make sure that it has enough of that life-giving oxygen. When we talk about blood, usually you hear blood type. That's kind of the first question that people ask you when you start talking about blood. What's my blood type? What's your blood type? Your blood type as an individual is inherited. You get a copy of an allele from mom and a copy of an allele from dad. The different alleles are, and the different, phenotypes, so remember phenotype is the physical expression of a gene, are A, B, and O. So A and B are codominant, meaning that if they are together, if you get an A and a B, one from mom, one from dad, you will have AB blood. O is recessive to both A and to both B. So you can have blood types of both where you have homozygous A or heterozygous with an O, with a recessive O, and same thing with B. So there are 4 different blood types. We're going to talk about plus and negative because I'm sure that's on the tip of your mind there. These are the 4 blood types and the genotypes that make these phenotypes. So even though we have 6 different genotypes, we only have 4 available phenotypes. So inside of this blood, what makes it this type is the antigen that it has on it. And antigens are the cell markers that are on side of cells that kind of say, hey, this is what I am. I am an A-type blood cell. And most cells in your body have some kind of marker on them that say, hey, I'm your cell. And so when cells from outside of your body come in, then we have a reaction with the antibodies. And antibodies are these things that say, hey, you're not from around here. I'm going to bind to you and allow the immune system to get rid of you. So type A blood is going to have A antigens. And the antibodies will be the things that it doesn't have. There's no B antigens on this, so it will make anti-B. It wants to get rid of these Bs. The B type blood will have the B antigen. It will make anti-A antibodies. So if A blood was to be introduced to a B type individual, they would have agglutination, or they would have that clumping of the blood inside of the vessels, and it would probably lead to death. We have type AB, which has both A and B antigens, so it has no antibodies. And then we have lastly type O, which has no antigens present, so it makes both antibodies. Now, when we talk about universal donor and universal recipient, universal donor is going to be O. Universal, let me use a little bit more remarkable color there. Let's use this green. Universal donor. And our universal recipient is going to be this type AB. Because AB has no antibodies, it's not going to react to any blood that gets transfused. Where type O, if it's not type O blood, if it's type A, type B, or type AB, it has these anti-A, anti-B antibodies, it will attack that, clot, and usually ends up being fatal for the patient. It's not great to have blood clotting inside of your body. The Rhesus factor, this is the Rh positive or minus. We call it the Rhesus factor because it was first discovered in the Rhesus monkey. And this is another genetically given thing that determines whether you have a plus or negative on your blood type. So A minus, B positive. This is due to two different genes, one for blood type and one for Rhesus factor. Now, Rhesus factor is really important when we talk about mom and baby. If mom is a negative and baby is a positive blood type, her immune system will want to create antibodies for that and will want to attack it. And so we need to use this Rh immunoglobulin, and this helps prevent this Rh, what we call sensitization. So somebody with a negative blood type can receive a, let's say, let me just real quick write this down so we can keep track of it. Somebody with an A negative blood type were to receive a transfusion from somebody with an A positive blood type. Because there's no sensitization already, there's no to this Rhesus factor, this transfusion would work the first time. That positive blood would go in there, it would do its job, but in doing so, it would alert the A negative immune system, and they would say, hey, let's make this antibody against this positive for next time we see it. And so that will happen, and then the next time, further down the road, that this A positive is then tried to transfuse, we will have that transfusion reaction and we will have agglutination form. So this Rh immunoglobulin prevents the sensitization process so that mom doesn't attack baby, which is very important. It's a very nice thing we have in modern medicine, and it's a very uncommon thing now. There's usually testing done very early in a pregnancy to see if this situation is going on. Now we're going to switch gears and we're going to talk about synthetic oxygen carrier, and then specifically we're going to talk about HBocs, which are hemoglobin-based oxygen carriers. And these guys are used to provide temporary support to the circulatory system until that bone marrow is able to restock and resupply the circulatory system with the much needed red blood cells. They focus on getting the blood from the lungs to the cells. And they have many applications. We could use them in cases of trauma, for elective surgery, use them for cardiovascular surgery, perfusion of ischemic tissue, for things like sickle cell anemia, individuals who've had strokes or peripheral vascular disease. And we can use them to preserve organs during transport for transplantation, so to keep these organs extra life. And primarily we would use these when we would use a transfusion, but we don't have whole blood available. Now these are derived from hemoglobin, natural hemoglobin, which has been isolated or synthetically manufactured. We'll use outdated human blood or bovine samples to get this. And oxygen will bind covalently to these compounds the same that it does to natural hemoglobin. And this on the left here is from Haller et al. It is this hemoglobin-based oxygen carrier that is synthetically made, and this is a normal hemoglobin molecule here. And as you can see here, they are very similar in structure and therefore should be very similar in function. They are going to help out with this need of not having enough oxygen in the cells during these times when blood transfusions are hard to come by. A kind of consideration that needs to go in these guys need to be these guys need to be filtered or heated to get rid of some of the viruses or proteins that might cause some contraindications. And they also need to kind of take into effect that these guys can expand plasma, so they can affect blood pressure, and affecting that might affect the intravascular volume and how much of it's in the system. Also, these guys don't last as long as red blood cells. So most of them will last 20 to 30 hours inside of the body, whereas a normal blood transfusion lasts 34 days. Like I mentioned earlier, our first choice for obtaining this is through outdated human blood, and the second is through bovine blood or cow blood. But the downside to collecting bovine blood is we can have the pathogen, the prion pathogen that causes Creutzfeldt-Jakob or mad cow disease inside of this. So there's this study. In the study, they talk a lot about using engineered bacteria to make a recombinant source for this human hemoglobin, but there's still a lot of work before we get to that point. But it is an exciting application of cardiovascular and circulatory health, and hopefully one that we can crack so that we have these substances to use when we don't have transfusions. Some references: the bulk of the actual anatomy and physiology from this presentation was provided through the textbook by Sherwood. There's a lot of great information on there. And then this Haldor et al., this futuristic dimension of modern-day transfusion sciences, includes this HBocs and a couple other methods that I didn't talk about that are very interesting. It's a good read. If you're interested in that kind of thing, I definitely encourage you to check this one out. I hope you learned more about blood. Red blood cells specifically, their structure, function, and their place in society as far as blood typing and transfusions, and some potential research for creating synthetic hemoglobin to carry blood to tissues that need that life-saving oxygen. Thank you again for watching, and have a nice day.